Hello and welcome everyone. Thank you first to Fullscript for hosting this event. We're very excited that MediHerb and standard process products are now available to practitioners who use the Fullscript website. I'll be exploring two key areas tonight. First, to celebrate our launch on Fullscript, I'll take some time to introduce you to MediHerb and talk about our history and our unique approach to herbal manufacturing. We will then move on to an all-important clinical topic, and that is of immune and respiratory health, and challenge some common misinformation about herbs in this area. Feel free to submit questions in the chat as we go along. We don't have time to address the questions today, but they will certainly be responded to afterwards. So this is a statement that I have made that still rings true today, even though I made that statement possibly more than 2 decades ago, that I and my wife co-founded MediHerb. I'm sitting here in Warwick, Queensland, Australia, and I'm not on site at the factory, but the factory is literally 2 miles that way. So MediHerb was born out of my desire for efficacious herbal therapy. And I truly believe even over all these decades, it still remains the driving force behind every aspect of our endeavors. And our passion is to provide optimum treatment solutions by combining the time-honored wisdom of traditional knowledge with sound clinical experience and the rigor of scientific research. And for example, I am still in busy clinical practice. I average 25, 30 consultations a week and have been doing so now for more than 35 years. So this is where it all began. So, you know, we know about large businesses starting in garages and what have you. So we went one better. We started in a goat shed on a farm. We did actually kick the goats out first and converted it, but that's where we began. And there was just my wife and I and another worker. We had no sales or reputation. We weren't even sure, you know, our business backers wanted us to be more retail, but I said to them, look, I know about practitioners, clinicians, herbal clinicians, and what they want. And I think our focus should be there because I think there's a real need to be met. And that certainly proved to be the case. So one thing I do want to emphasize is the MediHerb products are not retail. They're a professional range for healthcare professionals. Shortly after we began, we founded, and here I'm talking about 1987, the chap on the right, the man on the right, Peter Perbrick, joined us, and he is still here with us at MediHerb today, and he's involved in herb sourcing. And so we have that expertise and that continuity that's unparalleled with our herb sourcing. This is what the factory looks like at Warwick today. It's quite large, and you see bottom left, if you go look at that image, you can see as you go up to the full extent of the factory, there's a building which looks like it's got a bit of a black face to this side. That's all still part of the factory. So it's around about 90 people work there, and then we have offices in Brisbane. There's another more 10 to 15 people who work, probably even maybe 20 people who work in the quality control and quality assurance labs. You see we've got some lovely herbal drawings on our warehouse there down the bottom right, and it's no surprise you'll be seeing echinaceas there, which is my favorite herb, and one that I'll certainly be talking about today in my second presentation. So Peter Perbrick and team source herbs from all over the world. We have contracts with growers. We also source from major suppliers as well, wild crafters and so on. So given this extent and breadth of our herbal sourcing, naturally, you need to have excellent quality control procedures to make sure you are getting the best possible quality ingredient. And that's what we do. We have, as you can see below, a global quest for quality. And you can see that webinar on standardprocess.com. So as we know, species are not the same. Cats, dogs, fish, whatever. And certainly that applies for herbs. And with herbs, we have the added complexity of plant part. And this, for example, applies with a herb like tribulus because a lot of companies sell tribulus fruit, whereas the impressive effects of tribulus in terms of modulating hormonal function in men and women are actually for tribulus leaf. So, and it's quite interesting when I look at US labels of US herbal products in particular, it applies around the world, but it seems especially in the US, you'll find quite often they don't even state the plant part. That to me is shows that that there's something missing there in that company's understanding of what herbs do and the approach to quality, because plant part is hugely important. So we start off with our botanicals, and they have complex phytochemistry that gives actions and ultimately clinical indications. So we need to get that complex phytochemistry right. And herbs, of course, are inherently variable. You have issues with genetics, environment, how they were grown, what stage they are harvested. Post-harvest handling is hugely important, and all our suppliers adhere to what's known as good agricultural practice, which has been developed for herb supply, species, as I've mentioned, and plant part, and then extraction and manufacturing, and also finished product stability. And this is kind of a blind spot for most manufacturers around the world. They've made a great product, but is it stable for the lifetime of the product? Well, we have an extensive stability testing program. Every product we make, multiple batches are tested in incubators for their stability over the lifetime of the product, where we look at phytochemical profiles to see if there are any changes. And if we see any changes, we make adjustments either to the shelf life, or we might change what we put in the product to help it be more stable, or we might even put an overage in so that the small change in activity that occurs over the lifetime is more than compensated for. And that's particularly when we state that there's a certain level of a marker compound in the product. What we're stating is that marker compound is there at that level at the end of the lifetime of the product, not at manufacture. So we've been operating under conditions of pharmaceutical GMP, good manufacturing practice, for 3 decades now. And of course, more recently, when the FDA has decided to audit herbal manufacturers, we also have approval from them, and we have been audited by the FDA, I think now on 3 separate occasions. But we have this culture that's completely inbuilt of pharmaceutical good manufacturing practice. And because in Australia, herbal products are classified as medicines, we actually manufacture to a pharmaceutical standard. And at one point, because certain herbs like ephedra are classified as full drugs in Australia, we actually had a full drug manufacturer's license. That's how high our level of adherence to GMP. But because we no longer sell ephedra, we don't need that anymore. So as well as good manufacturing practice, good laboratory practice is also required for herbal products, and our laboratories are extensively audited by the Australian authorities. And it's documentation and documentation. It's all electronic now, but we used to have a joke in the factory that GMP stands for generates much paper, and the paperwork is extraordinary. And all critical steps, for example, in manufacture, adherence to line clearance is noted, so you don't confuse one product being made with another. Critical steps in manufacture are both certified and verified. So someone does it and someone observes them doing it and then certifies that they did it correctly. So all of those procedures are followed and documented. And we also need to get our products approved by the Australian authorities, and only once that product is approved is it allowed to be sold in Australia and overseas. So our manufacturing process, we start off with the raw material storage and our warehouse again there on the right. And you can see that it's we have humidity and temperature controlled, and it's kept at around 14 to 16 degrees centigrade to help optimize the storage of the herb. You don't want it too cold because you might get some condensation and other things like that, and it's not good for the workers. And so that low temperature of around 14 to 16 degrees centigrade, it's wonderful on a hot Warwick summer's day to go into that warehouse. We mill the herbs. We also have a herb cutter as well. So depending on the herb and its nature, we'll cut it or mill it. And the raw material is if the herbal actives or marker compounds like essential oils are temperature sensitive, we'll actually mill them under conditions of dry ice so that they're actually cold milled, and we don't see any losses in the milling. And then we still operate extract using a proprietary 1 in 2 cold percolation process to produce our liquid extracts that are the main liquid product we sell for practitioner formulation for the individual patient. So we don't use heat or kinetic energy. So it's not like a coffee percolator, and that's why we use the cold percolation. The term cold percolation, it's not that it's freezing cold in the percolation room, it's that it's 21 degrees centigrade. It's that we don't use heat in the process. It's a very slow process. It takes a week to 10 days and allows for a natural extraction of the full spectrum of phytochemicals. And a lot of companies make tinctures by maceration. Just, you throw the herb in, you throw some alcohol water in and just soak it and shake it for a week or two. Well, we use a slow percolation process, which is far better and more efficient for extracting. But because we do it over a period of 7 to 10 days, you get the kind of like the benefits of maceration as well. And then we turn those liquids into tablets. We do buy in some extracts from extract suppliers that are specialized, and they undergo highly rigorous quality checks. And the requirements of the US Pharmacopoeia are just our starting point. We have the phytochemical knowledge to understand the subtleties and do much, much further testing to ensure we're getting the right extract and it hasn't been adulterated. But most of our products, actually, the liquid is used to make the tablet. So that's unique, and there's no other company in Australia, and there's not that many around the world that can start with a herbal raw material and then go right through to a finished solid dose product, and we can make both tablets and capsules in-house. When we're making a tablet or capsule from the liquid, we use low temperatures to evaporate the water ethanol mix that's been used to extract the herb. And we have technology such as falling film evaporators and so on that do that. and that's all done under pharmaceutical GMP. So let's talk about our testing. Any raw material, first we do pre-shipment testing, and then when the batch comes in, we sample according to good manufacturing practice sampling procedures, and again test the raw material. Then if we make a liquid extract, we then test that either as a finished product or as an intermediate product, because it might go into a tablet or capsule, and again we test that tablet and capsule. And finally, as I mentioned, stability testing. So anything we put on our labels about what that product contains is actually validated at the end of the product life, not at the beginning. And again, we have seen that that's not always the case for other herbal manufacturers. And again, all done under pharmaceutical good laboratory practice, GLP. So that's summarized in this diagram here, or this figure here. Up the top, you have the processing, where you're going through sourcing, milling, extraction, and down the bottom you have all of the testing. So if we're producing a tablet product, for example, then it will be tested 5 times in the manufacturer, and then a 6th time for its stability. So what are we testing for? Well, obviously the identity and the validation of the species and the plant part, the phytochemical profile and the levels of key marker and active compounds, and I'll talk more about that soon. We do it both quantitatively, looking at the full spectrum, qualitatively looking at the full spectrum, and quantitatively looking at amounts of certain chemicals. Adulteration and substitution are key things that we look for. And we're very sophisticated, and we have an understanding of the tricks that suppliers do, plus many other factors that relate to this, like microbial levels. We have very strict microbial requirements on our products from the Australian TGA. That doesn't tend to apply much under US FDA. We look at aflatoxins, heavy metals, foreign materials, infestation, pesticide and herbicide residues, solvent residues if it's bought in extract, moisture levels, and so on. And something that we're testing for regularly now, because it is on the horizon of regulators, and it will become, I think, a significant issue for the industry, and indeed problem going forward, is we test for pyrrolizidine alkaloids. So what happens is that if the herb that you like is harvested with a weed, and that weed contains pyrrolizidine alkaloids, well, then you have a toxic plant contaminant in the product, and we are testing for that as well. And I think very much being world leaders on that, and of course stability. We know that contamination is a problem, and the US FDA has highlighted a number of issues. It's found hundreds of dietary supplements, for example, adulterated with prescription medicine, and even after FDA recalls, the company seems to continue to supply them. There's been examples of mycotoxin contamination, for example, milk thistle. And the reason why you get mycotoxin contamination for milk thistle is the part used is a seed, and it's a fatty seed. And as it dries, it can get a fungus on it if it's not dried properly, because the fungus loves the fatty oil that's in the milk thistle seed. Heavy metals have also been shown in products, and in the US, and pesticide residues. And don't forget, if the company is using bought-in extracts, there may well be solvent residues. Well, that's never an issue, because even if we buy in an extract, we now have guidelines that alcohol, ethanol, and water should be the only solvents used for any new extracts we buy in. So here's the degree of botanical testing we do. We do gas chromatography, high performance thin layer chromatography, and ultra high performance liquid chromatography. And for UHPLC, PLC, the last thing I said, you will use different techniques for detecting the phytochemicals that are in the herb. And one of the techniques we use is cutting edge mass spectrometry, as well as the more standard ultraviolet visible diode array and light scattering and so on. So our lab manager, who's been with us for quite a while, Aaron, says our stringent testing regimes guard against substitution, adulteration, and poor quality. And that is exactly what they do. We have a range of in-house phytochemistry experts. We have R&D scientists, method development scientists, quality control scientists, and managing scientists that drive innovation, and they interact with the MediHerb research team. And we also have cross collaboration with a range of outside experts. So we're doing extensive research now into beta glucans in mushrooms, for example, and we've published on this. And the complexity of quantifying mushroom products and avoiding adulteration are huge and not very well defined. So we even have a number of PhD projects going on this. We have several PhD projects, and we're in constant communication with expert centers at universities and other global experts and associations. And this is these are our expert team, and you see it says Dr. David Leach there, but David has been a full professor of phytochemistry at Australian universities and is recognised as one of Australia's leading phytochemists. Dr. Reg Lehman is also an associate professor at the University of Queensland, and Dr. Hans Woolmuth is an associate professor adjunct at Western Sydney University. So you see it's professor, doctor. But they are indeed experts, and we're blessed to have such a wonderful senior team. I am blessed to have that I collaborate with on both research and quality. So our pioneering leadership in botanical research and development has been recognized. For example, the American Botanical Council awarded us the Varro E. Tyler Award for phytomedicinal research, and said that MediHerb is a company that invested millions of dollars to back up the benefits of herbal medicine with sound science. And in my second presentation, I'll be talking about some of that in the context of Echinacea, our Echinacea research, and also the industry body in Australia awarded us with an outstanding commitment to R&D award. As you can see, what that's based on. So this slide summarizes what I believe is the MediHerb difference. We have botanical sourcing expertise, unique proprietary manufacturing processes under pharmaceutical GMP, unrivaled testing expertise under pharmaceutical GLP. We have a group of unique IP leaders in research, and we have the best and talented people doing our education and our marketing. And importantly, we have clinicians on staff who are in clinical practice. I'm in clinical practice, and so most, if not all, of our educators are still in clinical practice or have had many years in clinical practice. And that also comes through, I think, in the way we formulate products. So we have a range of tablet products, and what I say to clinicians is we have herbal expertise that's built into the product. We don't fairy dust products. We don't think of every herb, for example, that's good for prostate health and throw it in there, but think, oh, it's going to be too big and too costly. Hang on, we'll just put these 3 herbs in at 50 milligrams, 100 milligrams, 10 milligrams per dose. That's called fairy dusting. We don't do that. We design clinically relevant and effective products. And what this means for both the clinician and the patient, that you're getting a quality, efficacy, safety, and above all, I think, consistency. Because the last thing you want to know is if a patient comes in and they say, oh, that product you're giving me doesn't seem to work as well. Last thing you want to know is that batch is so different from the other one that you gave them before that worked. You want that consistency from batch to batch, and that's what our sourcing and testing, the whole box and dice, that's what that guarantees, including stability. It guarantees that consistency. So it was a company, MediHerb was a company that was established by practitioners for practitioners. And for over 30 years, we've been passionate about investing in the future of natural medicine and delivering innovative healthcare solutions that provide the healing wisdom that patients are seeking. We're not a fledgling startup. We are well established in both the Australian and US markets. And we have a substantial turnover in the US. Probably more than half of what we produce at that factory here in Warwick goes to the US. So if you're not currently using MediHerb products, please do consider them, and you'll find that they will give you the clinical results that I've certainly built my practice on for the past 35 years. So thank you, and I'd like to acknowledge, and with my appreciation, the various contributors to this presentation, some of whom I've mentioned along the way. Let's move on to part 2, my second presentation. And here I'm talking about herbs for maintaining immune and respiratory health. And our topics will be first talking about my absolute love of Echinacea root and understanding its critical value, because I certainly believe it's still a herb that is underestimated. Talk about some other key immune herbs that I believe work well in clinical practice. Just briefly touch on this topic of immune herbs and cytokine storm risk, because you do have an associated paper that I've written that explores this much more fully. And I'll talk about antiviral herbs, and here I will emphasize it's important to use ones that are clinically relevant, and I'll explain what I'm meaning there. And then just some core protocols about, firstly, about just how we can support immunity and resist the various challenges to the body. So key immune herbs, and of course, Echinacea is number one. And what I want to stress here is that the herbal approach to infection, given the context of what we're seeing now with various issues, is we are approaching a crisis point in having a need for agents that help control infection. So what can we offer? What's the key herbal action when treating or preventing any type of infection? It's immune support. I believe that is our strength, and we need to play to our strength. And I used to see a scenario when I was teaching undergrad, and that I'd ask about a particular condition, and the students come back and say, oh, I'd use this herb because it might be antiviral, or this herb because it might be antimicrobial. And despite my drumming into them, they'd miss putting immune herbs in there. That is actually the strength. Immune support is core to any herbal approach to any kind of infection. It's our frontline treatment, and it's fundamental. So what are our key immune herbs? I've classified them as major. So Echinacea and Andrographis, I believe, are the major immune herbs. And then other immune herbs that help. You can make a longer list of others, but I've put in examples of holy basil and adaptogenic and tonic herbs, but especially astragalus. We can put cat's claw in there as well, and of course medicinal mushrooms. But I see Echinacea and Andrographis as as major. And Echinacea indeed is my favorite herb, and I believe it has much to offer the modern herbal clinician. But it is so confused and misunderstood, and we'll briefly be touching on this. And you have a second handout from me, which is the Echinacea root monograph I wrote for the second edition of Principles and Practice of Phytotherapy. So I've given you that in PDF form. And many of the issues and myths and confusion and misunderstandings that I touch on here are fully explored and referenced in that document. So if you don't have that book, or if you do have the book, go and read that monograph. But if you don't have the book, you should. But if you don't have it, then the PDF is there for you to look at. So part of the confusion of Echinacea is we have a multitude of products using different species, plant parts, manufacturing methods, and confusion over what phytochemicals in Echinacea are responsible for its immune activity. And only I think at this stage a basic understanding of its exact action on the immune system, but with some intriguing recent developments and many myths about how it should and should not be used. Now, all of these issues are addressed in that monograph that you have as a handout. We can do no better in understanding Echinacea than to begin with where we learnt about it. And the information about Echinacea and its therapeutic value first came from the Native American tribes. Their use of Echinacea was then adopted by the eclectic physicians. The eclectic physicians were an important natural therapist movement in the United States from the 19th to early 20th century, and they wrote some wonderful textbooks and had their own journals, and they were wonderful clinicians. And it's interesting that by 1921, and that Echinacea, and specifically the root of Echinacea angustifolia, extracted with alcohol and water, so it was a lipophilic or fat-loving extract that had the tingling alkylamides, was by far the most popular treatment prescribed by the eclectic physicians. And this ad from the Lloyd brothers illustrates that. So you can see there's Echinacea, and look at the next most popular herb is Chiananthus, which is fringe tree, Macrotis, that's the old-fashioned name for black cohosh, and then Crataegus or hawthorn, Pulsatilla pasqueflower, Gelsemium, and Thuya, and Phytolacca. Now, you see many of them are sort of immune and potentially antiviral herbs in there, but look how Echinacea excelled. It's like as much as the next three combined. And that's despite the fact that it was only in 1887, so only 34 years prior, was Echinacea introduced to the eclectic physician profession by Dr. John King, and only in 1890 did the Lloyd brothers start making that alcoholic extract of it. How the eclectics used Echinacea, it is discussed somewhat in the Cytokine Storm paper, but I suggest you have a look at the monographs in Felter and Lloyd and in Allingwood and Lloyd, and they're both available online. They also have been, if they're still available, and I have copies of these reprinted by the Eclectic Medical Publications, and it will blow your mind away reading through how they used Echinacea root and the clinical results they obtained with it. And the underlying theme from these uses is immunity. It's immunity. You can see that even though at the time they were writing those monographs, they had barely any understanding of the immune system. So I can remember I was actually in the United States doing a round table with a group of naturopaths, and I was giving my pitch on Echinacea and how we should be using the root and the high alkylamide preparations like the Eclectics, because, you know, there's a lot of talk, oh, it's the polysaccharides that make Echinacea work, so you want to actually extract them with low alcohol or even use a pressed juice or things like that. That's a later European adaptation. It's not something that was ever done by the Eclectics or the Native Americans. and often, they're using the above ground parts because, hey, there's more polysaccharides in the above ground parts. Well, that's a great commercial advantage to companies doing that, because that's very cheap compared to obviously getting the root. And I was saying, no, you need to use the alkylamide, high lipophilic alkylamide preparations. You need to give substantial doses. It's all backed up by what the Eclectics wrote, because I was so influenced by what they wrote, and then by taking that and applying it in my clinical practice to huge success. And they challenged me quite fairly. They said, well, it's only traditional. You're just talking about your clinical experience and traditional knowledge. Where's your evidence? So that's where we began our journey to generate that evidence. And you can see I'm a co-author on this paper looking at the pharmacokinetics of Echinacea root. So we did a proper study in a pharmacokinetic lab at a hospital in Australia, 11 healthy individuals aged 18 to 26 years, and we gave them 4 Echinacea root tablets as a single dose and monitored their blood over 12 hours. So what did we find in their bloodstream? Well, none of these caffeic acid derivatives, we didn't find those. We didn't even find their degradation products. We didn't find degradation products of alkylamides, and we certainly didn't find any polysaccharides that could be attributed to the Echinacea, I guess because there was none there because we were extracting with alcohol and water anyway, but polysaccharides are not really bioavailable as such. The only compounds we identified in the human plasma were alkylamides from the Echinacea ingested in approximately the same ratio of the initial product tested. So you don't need to be Einstein to join the dots, do you? If this herb is influencing immune cells and immune cells throughout the body, it's got to be acting by agents that get into the bloodstream. And the only agents that get into the bloodstream when you take a traditional Echinacea lipophilic root extract are the alkylamides. And the Eclectics were no fools. They said, when chewed, the root, if of good quality, imparts a persistent tingling sensation. And that's the quality test for the alkylamides, the organoleptic test you can do yourself. So we now have a much better understanding, and you can see that in the monograph I provided you as a handout. We now have a much better understanding of what the Echinacea alkylamides do. And we've been involved in some of this research. Some bind strongly to cannabinoid receptors and cause a subtle modulation of immune function. Yes, you heard me right. Cannabinoid receptors. We have all this craze, don't we, about cannabidiol and what have you, yet the Echinacea alkylamides bind more strongly to cannabinoid receptors than CBD does. And we believe that leads to a modulation of immune function and anti-inflammatory effects. Other alkylamides might inhibit the breakdown of your endogenous cannabinoids, your endocannabinoids, and then make them more effective at those cannabinoid receptors. There's possibly an upregulation of dendritic cell maturation, which are involved in immune surveillance, and there's priming of innate immune responses and support of natural killer cell numbers, activity, and viability. So this highlights a very important point, and that is that what we seem to know now about alkylamides and their interaction with cannabinoid receptors and the other pharmacological models that have been used, that Echinacea seems to be acting more on immune vigilance and early innate immune responses. Now, if that is the case, then obviously it should be taken for prevention more than anything else. And that's how I've applied it in my clinic. I kind of accidentally stumbled on that, both through personal use and in the clinic. And it's been tried and tested now for decades, not just by myself. And it really does work as a preventative agent. That is actually its strength. And another important thing, of course, is that our innate immune system ages, and particularly the natural killer cells decline in number and function with age. And it's thought to be a factor behind the increase of various diseases with age. And Sandra Miller from Canada, her experiments in healthy elderly mice found that 2 weeks of oral doses of echinacea root returned natural killer cell numbers and function to levels that you see in the young adult mice. So it's kind of a rejuvenation of the immune system. And in addition, as a result, mice fed Echinacea purpurea root from 7 weeks of age to 13 months, their lifespan was significantly extended. So my hypothesis and my personal use of Echinacea is it's an anti-aging herb. And as a result, I take it every day and have been taking it every day for 25 years. Let's talk now about Echinacea in terms of clinical data. And this information won't be in your monograph because it postdates the monograph preparation, which was around about 2012. And we have a Cochrane collaboration assessment from 2014. And they make a very important point that hadn't been really highlighted in publications to this date, although, of course, we've been saying it for some time, that most consumers and physicians are not aware that products available under the term Echinacea differ appreciably in their composition. They assess the clinical evidence, the evidence from clinical trials that Echinacea preparations are effective and safe compared to placebo, and the models have been mainly used in their trials with prevention and treatment of the common cold. Of 7 treatment trials, only 1 showed a benefit for Echinacea. Most prevention trials were inconclusive, but when they did a post hoc pooling of the results, there was a relative risk reduction of 10 to 20%. So they did a post hoc pooling because of the, rather than a proper meta-analysis, because of the fact that Echinacea differs appreciably in its composition, the products that were in the trials. So there's a suggestion now, even from the clinical evidence, that Echinacea is more preventative. And this was highlighted by a study, as you can see. Oh, sorry, this is the Cochrane one. And this is the prevention summary on this figure from the Cochrane. And for example, the clinical trial we did is there. And you see, that's the only one for which the error bars, in this case, P equals 0.05 rather than less than 0.05. So that's why the error bar is touching the line. You see, for all the other trials, the error bar goes over the no effect line. And so none of those trials were statistically significant. Ours was touched on statistical significance. And this was a prevention trial, our trial in air travelers. But look at the average point, the little horizontal blue dot from all of those trials. In general, it's all to the left, isn't it? So what does that suggest? It suggests overall the data is looking like it is a preventative, despite the confounding effect of various preparations. And this was highlighted in this 2015 meta-analysis. And they concluded the following, and it's quite strong conclusion, that the use of Echinacea extracts is associated with a reduced risk of recurrent respiratory infections. In other words, preventative use, the relative risk 0.649, so 35% risk reduction. And that's across all products, and some better quality than others, if you get my drift. And ethanolic extracts, hey, from Echinacea, the more traditional lipophilic extracts, appear to provide superior effects over pressed juices, and increased dosing during acute episodes further enhance these effects. So that completely validates the approach that that I, for example, have been advocating about how Echinacea is best used. Now, some of you may have, had a shock reaction when I said I've been taking Echinacea continuously for decades, because you may be thinking, well, Echinacea root, you can't take it long term because it wears out your immune system. tachyphylaxis. So I've done some forensic analysis of this thinking. I was very confused when I first heard it, and it's quite interesting. I heard about it a couple of decades ago, which is just at the time I was concluding that the best way to take echinacea was continuous use. And I believe it comes from this study. So what you see here is it was an echinacea root tincture given at 30 drops 3 times a day versus placebo. And they looked at one immune function of the white cells in the bloodstream, which was phagocytosis. So the first thing to say is they only looked at one aspect of immunity and not one that's preferred in modern models these days, which was phagocytic activity. That's the first thing. Now, if you have a look at the graphs, you see the darker blue line, it just sort of goes along the baseline. There's no effect from placebo. But you look at the echinacea effect and you see, yes, it's boosting phagocytic activity after 5 days, but then it suddenly reverses after 5 days. And this has led to the thinking, oh, you can only take echinacea for 5 days or a week or so before it starts to wear out your immune system, this tachyphylaxis. But this was, as you can see, this paper was published in German. And so when you're translating from the German, you can miss things. And what was missed, and is clearly shown by the arrow bars below, is that day 5 was a day they gave the last dose of echinacea. So what you see after day 5 is not a wearing off of the effect of continued use of echinacea. It's a standard therapeutic washout phenomenon. You stop the treatment, so its impact reverses back to normal. Now note, reverses back to normal, not below the baseline. So however, this thing that you're going to be subpar with your immunity with continuous use of echinacea, where that came from completely baffles me. And it's also quite the opposite, because if you look at day 8, there was still around about 35% residual impact on phagocytic activity. So it had a lasting effect. So anyway, more fully discussed in the monograph. Let's move on. And you can see even at day 7, there's a substantial residual effect. So Andrographis. Andrographis has good evidence in winter infections. So Echinacea more for prevention, Andrographis more for the challenge. 2 systematic reviews have been published. In one review, looked at 7 double-blind controlled trials in nearly 900 people, and the trials weren't of great quality. We don't tend to use the JADAD scale anymore. We use the Cochrane risk of bias, so all trials did score at least 3 on that scale, on the JADAD scale. And the data suggested that Andrographis was superior to placebo in alleviating subjective symptoms of uncomplicated upper respiratory tract infection, and provided evidence also from one trial of a preventative effect. The second review looked at 3 trials in the meta-analysis, and Andrographis either alone or in combination with the Herbal Eleutherococcus was more effective than placebo in the treatment of uncomplicated acute respiratory tract infections. Now, it's important that we dose adequately. They were typically 1,000 milligrams a day of extract, which is about 6 grams of herb, and delivering about 50 to 60 milligrams a day of andrographolides. And that's what I use to guide my dosing under these circumstances. Now, Astragalus is good for building the immune machinery, and that's what's been shown in this trial. So we're going to use Astragalus for people who are particularly depleted in that regard. So there was a randomized controlled trial, 150 patients with low white blood cell count, received 2 different doses of Astragalus, and there was a significant rise of white blood cell counts in both groups after the treatment with Astragalus. And there was a dose response effect seen. So for the higher dose, you saw a much greater rise. Now, if we look at China and recent experience they are using Astragalus and Licorice in many of their formulations now to prevent infection. So I mentioned I have a supplementary paper for you on immune herbs and cytokine storm risk, and this seems to have come into the attention, especially online. Concerns have been raised that certain herbs acting on the immune system might deleteriously enhance the cytokine response during acute respiratory viral infections and lead to cytokine storm, which is otherwise known as sepsis. But they're all based on a fairly narrow one-dimensional analysis of test tube studies, and they're not supported by a detailed analysis of the published scientific and traditional literature, which is what I provide you in that paper. And also, more importantly, they don't differentiate between the initial role of cytokines during an infection as immune signaling agents versus their much later role in late-stage infection in promoting the inflammation of sepsis. And interestingly, Allingwood regarded Echinacea root as the herb for sepsis, which we call cytokine storm. And again, that's documented well in the paper. So since the main roles of Echinacea, Astragalus, and medicinal mushrooms are for prevention, the argument behind their concerns over the use of cytokine storm risk is made even weaker. And cytokine storm is a chaotic, intense, and unregulated response to a massive necrotic tissue destruction late in an infection that's unlikely to be capable of any further augmentation by any agent, much less a relatively benign medicinal plant. In fact, what immune herbs will do, though, is they'll support a focused initial immune response, including, as I mentioned, enhanced cytokine signaling, and thereby reduce the risk of any infection progressing to the stage 3 development of cytokine storm. So now move on and talk about some key herbs that have antiviral activity. And here I would like to inject a note of caution, and I call that note of caution in vitro non veritas. In other words, you don't necessarily get the truth from in vitro research, and that particularly applies to people trying to extrapolate test tube research about any agent that has antiviral activity in a test tube versus whether it might be effective in a human being. So the list I'm giving you now, I believe, is a credible list based on an in-depth analysis of the data that includes both animal and human data, and doesn't overly rely on test tube research. And the important point is, if you are relying on test tube research, you need for that plant chemical or the key components of that herbal extract to be highly bioavailable. Because if they're not, and we now know there's this incredible cost talk between our gut microbiota and phytochemicals, whereby the microbiota changes those phytochemicals substantially, and itself is changed in that process, in terms of the relative levels. We have a high risk that in vitro non veritas will apply to phytochemicals because that extra factor of microbial interaction and change. So the main, if we look at Artemisia annua, it's phytochemistry. We're mainly looking at the Artemisinin derivatives, but it also, and the Artemisinin is found in the glandular trichome, so it's easily damaged and it's quite an unstable compound. So you need to be doing very rigorous quality control. And I'm going to tell you something that you may be unaware of, that Artemisinin is so unstable, it does not exist in liquid herbal preparations. It will have decomposed well before that product gets to you and sits on your dispensary shelf. So if you want to get effects from Artemisia annua that are Artemisinin related, you need to use solid dosage forms, tablets, capsules. we, of course, look at the whole plant, not just the artemisinin, and it contains flavonoids and essential oils, and they're all important. So what you see here on the structure of artemisinin is this peroxide bridge. And that's what confers its pharmacological activity, because that double bond there, if I show you with my mouse, see that that single oxygen-oxygen bond, I should say, that bond breaks and you get two peroxide radicals, and that's what confers its pharmacological activity, but it also confers its great instability. So you need solid dosage preparations that have stability that's been assessed at the end of the life of the product. So artemisinins have reported potent activity against viruses in vitro, herpes viruses, cytomegalovirus, Epstein-Barr virus, and there's human data for cytomegalovirus, human clinical data. And then hepatitis viruses B and C. We know that artemisinin is bioavailable, so we can extrapolate. As long as we get the dose right, we can extrapolate. And indeed, there is a human study that showed it was active against cytomegalovirus, an artemisinin derivative. And it has quite broad spectrum antiviral activity, as you can see from this table, against a range of classes of viruses. So the black squares denote where strong activity has been reported. Now, most of this is in vitro, but as I said, we do know that artemisinin is bioavailable. But another important point there, artemisinin is only bioavailable for a period of time. And that's around 7 days, because if you continually dose with artemisinin, you induce hepatic biotransformation, you induce your hepatic cytochrome P450 enzymes, and they then rapidly clear the artemisinin at first pass. So in the pharmacokinetic studies that have been done, you get good levels up to about day 5, and then they decline rapidly. And after day 7, there's virtually nothing there. So when I'm using Artemisia annua, I use it in pulse doses, a week on, week off, to allow those cytochrome P450s in the liver to downregulate, and then a week on again. So licorice, there are multiple studies show in vitro activity for licorice. Glycyrrhizin, which you can see the diagram there, but more importantly, it's a glycone, because this is a case of bacterial metabolism of the glycyrrhizin, makes the aglycone, which then is absorbed and then goes to the liver and gets phase 2 conjugation. And these phase 2 conjugates of the aglycone of glycyrrhizin do have activity, and that activity is against both naked and enveloped viruses. So it's like artemisinin, it has broad-based activity, and that has been demonstrated in animal models. St. John's wort, we know that hypericin has significant in vitro antiviral activity, but it's only against viruses that have an envelope. And it was actually originally researched as an agent for HIV infection. We do have clinical trials for St. John's wort in viral issues. There was one cohort with orofacial herpes and the other with genital herpes. And they took a baseline day of St. John's wort, and they doubled the baseline dose, I should say, of St. John's wort, and they doubled that dose during skin outbreaks. And the average symptom scores versus placebo were much lower in both trials, and there were also fewer outbreaks. Now, curcumin until recently is not something I had considered as something with antiviral activity, but I believe it does have clinically relevant antiviral activity. Firstly, there have been quite a few in vitro studies indicating broad-based antiviral activity for curcumin. And if you're using a bioavailable curcumin, which I do, of course, the bioavailability problem is addressed. Activity against HIV was recently reviewed. I'll show you that some of the findings there on the next slide. But it also shows activity against a range of other viruses, including in animal models, in this case against influenza A virus. So if we have a look at the curcumin and HIV, you can see it has several points of impact against HIV, which is fascinating, and it speaks to a concept that's now being talked about in herbal pharmacology, and that's this concept, and the Chinese have taken it up and done some incredibly sophisticated work with it, which is network pharmacology, and that's what that's doing. You're getting small effects on multiple targets, which could translate potentially into a big clinical effect, and that's what network pharmacology is about. And that's just for one phytochemical, and when you've got multiple phytochemicals and they're also interacting in the same network, that's why you can see that a herb that is mildly is pharmacologically mild can deliver a big impact through this phenomenon of network pharmacology. So let's finish off by talking about clinical protocols. So for general immune health, you can take Echinacea regularly. You can take it regularly. Someone said, when I said, look, you can take Echinacea regularly, I do it, been doing it for, 2.5 decades. Someone said, oh, but that's only an N of 1 trial. But believe me, I've witnessed it many times with patients, friends, family, and other colleagues have told me they do have the same with their patients. So it's quite safe to take continuously, and it does confer benefit. And as I said, I think there's a certain anti-aging aspect to its continuous use. Or if you prefer, you can use the Echinacea combined with Andrographis and holy basil. That can work quite well too for general immune support. If you think there's an issue with challenges about, you can also preemptively take these herbs, St. John's wort, licorice, thuya, which have some antiviral activity. If the challenges come through the throat, then you can use a herbal throat spray, and I do that. And for people who have a bit depleted, they need extra vitality support, then you can throw in some adaptogens there as well. So if someone is in a challenging situation, or they've come into contact with someone who's been immune challenged, you can temporarily double or triple the dose of Echinacea. I sometimes go even higher. And if you start dosing high with Andrographis, just watch for what can be a rare allergic reaction. I've never seen it clinically, but other people have. But it only seems to occur with a high dose, very, very high dose Andrographis product that was being retailed in Australia. But it has been documented elsewhere for high-dose Andrographis products as well. So just watch for that allergic reaction, and you can certainly switch over to Echinacea if that happens. Other options to help when the person is under challenge is adrenal support, and you can use these various options like licorice and Romania or rhodiola and schisandra. And if we look at the respiratory tract itself, at the very first signs of immune challenge, you can take until these signs of immune challenge in the respiratory tract resolve, and for a further 24 hours. So this is where that Andrographis Echinacea holy basil combination excels in my view. And you take it, acute issues, you dose acutely. And so you up the dose. Again, if you want to support the body's defenses more, you can use the St. John's wort, licorice, thuya, and we've discussed St. John's wort and licorice. Myrrh is also a wonderful immune herb, and I believe it's completely underestimated. And I invite you to, if you have a copy of Principles and Practice of Phytotherapy, have a look at the monograph on myrrh. It is so impressive. And Artemisia annua can also be taken, but as I said, for up to 7 days off, and then repeat if necessary. Other things you can do: garlic is often a great option, golden seal, mucous membrane support. I was taught traditionally that garlic is the tonic for or tropho-restorative for mucous membranes. I do have a new book out on functional herbal therapy, and one of the key precepts of functional herbal therapy is to support the tissue, and a golden seal supports the mucous membranes. And when we have some specific health challenges involving the lower respiratory tract, we can use herbs that support the normal mucus flow in the respiratory tract, and I've given you a list of those there. And we can, if there is a strong challenge and the body temperature is being threatened, we can support the normal body temperature by drinking diaphoretic teas. And I'm a big believer of diaphoretic teas, and you can also use formulations with pleurisy root. And again, if you look at that paper on cytokine storm that's available to you, I talk about the very important role of diaphoretic herbs. It's traditional, but it's incredibly powerful. If the throat's involved, a liquid herbal throat spray can be great to help soothe it. And again, because this can be a stressful situation, we can support the adrenals. And you can consider about turmeric if the immune response indicates that you want to balance it out and support a healthy inflammation response. So further respiratory support involves 2 formulations. I've given you that formula, which is more for immediate challenges earlier. And if you're using a liquid version of that, you put it in warm water because you've got the pleurisy root diaphoretic in there. And then there's this second formula that's more useful for after effects and dry hacking cough, and supports the normal mucus flow. That one you don't necessarily need to put in warm water. I'm not sure how that got in there, but anyway. Plus, obviously, immune support. So thank you so much for listening to this presentation. If you want to look at further information from me, there are my textbooks, including the new one. That's my 7th textbook for herbal clinicians. And you can follow me on Facebook. I've finally come to the modern age, and I have a Facebook page, and there's lots of interesting features on there, including Grumpy Old Herbalist, which is very popular. And with my co-author Simon Mills, we've also set up the Mills and Bone Academy, which is doing some very left field and groundbreaking exploration of clinical topics via the Mills and Bone Academy. So thank you very much. And don't forget your questions, put them in the chat box, and they will be answered. Not today, of course, but they will be answered.